Okay, thank you. I'm honored to be here to present on the topic of in vitro analysis of clot properties and their significance in mechanical thrombectomy. Thank you, Louise, for inviting me today. I started my career in biomedical engineering and was in R&D and quickly realized that I was very interested in medicine, so I completed a medical degree and wanted to combine my two passions and found Neuravi, which is a mechanical thrombectomy company based in Ireland, and they're very unique because they do a lot of fundamental clot research and analysis in their bid to create a thrombectomy device. So I don't practice medicine. I'm not treating patients like you are, but in our lab we have in vitro models in which, as a team, we've probably done more than 5,000 clot retrievals under 30 different anatomical models, and we use real blood clots to do that. And it gives us a very interesting perspective using visual light, not fluoroscopy, to get a really good amount of detail on the clot and its interaction. We routinely see in our in vitro modeling that different clots behave very differently. And in this presentation, I'd just like to briefly explain how we make our clots and some of the relevant properties that we have found and their potential impact in mechanical thrombectomy. Our motivation, of course, is to understand clots, to be able to recreate them and make a really, really good stroke model on the bench so we can prototype different devices and understand what works well, and then be able to test with different clots, different techniques, and how to use them most effectively. Even for someone like me who's just finished med school about over a year ago, the advances in stroke are just amazing. And when we look in the data, there's still definitely room for improvement on those patients that don't achieve recanalization. And while we know that TICI seems to be related to MRS and good clinical outcome, there are reasons why perhaps even those patients who have clots retrieved may not do so well, and sometimes patients cannot have clot retrieved. And these challenges may be from access difficulties, comorbidities, ischemic changes that might occur, and clot characteristics, which is really the focus of this talk. When we look at clots, just even visually from the literature, I find it just amazing to see these characteristics so differently. Clots that are involved in stroke can come from so many different pathologies, from so many different areas, and visually it's nice to see, or it's very interesting to see white, red, different shaped clots, different sizes, lots of different characteristics. So we couldn't figure out, or we didn't want to create a one clot fits all type of something, and so we created lots of different clot analogs that tried to mimic these different clinical clots. This publication in 2016 from our group shows how we make our clinical, or how we make our animal model clot analogs. We do this by looking at lots of different variables, and one of them is histology, specifically the composition of clots from stroke patients. And so we use that data along with other things like making different environments and aging the clots in different ways to create and develop these clot analogs that resemble human clots in their compositional nature under histology. And of course they're homogeneous because in vitro, but that really allows us to look at the properties in great detail. So with all the clot analogs that we've developed, we've learned a huge amount from investigating the material properties and behavior of these different types of clots. And one of the ways, as engineers, what we want to do is measure and quantify these different material properties to really better understand their behavior. So we've looked at properties like friction and resistance to indentation, compression and tensile strengths, and their fragmentation ability. But today I want to briefly describe our findings from studying friction. This is a very simplistic diagram showing a clot lodged in an occlusion experiencing pressures proximally and distally from the vascular bed resistance, and frictional forces between the clot and the arterial wall. All of that has to be overcome in order for the clot to be retrieved. So in studying the frictional property, we devised a simple experiment to look at that property. And basically we put a clot on top of a tilt table and then tilt the table until the time when the clot starts to slide, and the tangent of that angle gives us the coefficient of static friction. We've tested it on a variety of substrates, including bovine aorta, to find a polymer substrate that gives similar results, so that we can use a polymer substrate again and again and do lots of testing on it. So in this case, you can see a red cell rich clot and a fibrin rich clot, kind of both on the extreme ends of the spectrum. And in this video, we see them in a race. And what's really amazing, I think, is that not only are the angles so vastly different, of course these are in vitro clot analogs made on the extremes, but this friction-rich, the fibrin-rich clot, when it begins to move, it's even moving very, very slowly. So it really exemplifies the role of friction. And this timing of the presentation is very nice, I think, because just last week our group had this publication on clot friction in JNIS showing our analysis. And what we basically find is a significant difference between the fibrin-rich clots compared to the red cell-rich clots in their coefficient of friction. And looking at the trend analysis shows that there seems to be a threshold possibly around the 20% mark where friction really begins to increase almost exponentially. Furthermore, when we tested these clots, we realized that after every pass in our in vitro models, they seem to change shape and size as well. So we investigated what their impact would be if we compress the clot. And what we found was that whether we used a red cell-rich clot or a fibrin-rich clot, that compressing the clot seemed to increase the coefficient of friction. So the act of compression probably tend to dehydrate or shrink the clot, making it firmer and increasing that coefficient. It was very difficult to really grip anything without exerting some sort of compressive force, but this data does suggest that the less compression we have, probably the better. So one of the early observations we made was that certain types of clots appeared to require a higher retrieval force to be able to retrieve them. So we devised kind of a simple experiment to investigate this further. And here we have a really simple, well-collateralized vessel with fluid flow and the same setup and the same prototype stent retrievers being used. On the top is a red cell rich clot, or kind of a clot with about a 40–50% hematocrit, and it removes the device is able to remove the clot as we expect and probably hope in each case, maintaining the control. So not really much interesting to see there, but on the bottom video, we've replaced it with a fibrin-rich clot, and it's very interesting to see the stent retriever struggle to try and remove it. And not only does it not remove the clot, but it's also changed the shape of the clot a little bit. So if we look at snapshots from before and after the clot, we can see it shrunk in half almost, and that's probably compressed the clot, and as our testing showed, that compression probably increases the coefficient of friction. So this clot in the second pass may be even more difficult than the original attempt to remove it. We've also tested other types of clots, for example, to resemble clots that may have TPA loaded on board, for example. We've created clots that have a higher red cell content and may be more friable than others, and these clots are prone to fragmentation. So we were really curious to know what could be what could be done to kind of mitigate this risk, as there may be scenarios where we need to use, or you need to use, an intermediate catheter. And so in both of these videos, we see the same fibrin or the red cell rich clot that's tend to be more friable break up in different situations. One of the most interesting benefits, I think, with in vitro modeling is the ability to give a different perspective, a very visual one, and allowing us to see what's happening in a way that's not really possible in an angiogram. For example, these are two thrombectomy cases which both fail in quite different ways, and we'll play the videos. In the top one is a stent retriever, is a prototype stent retriever removing this heterogeneous clot, and in the bottom one, it's being removed through direct aspiration. And in both cases, the clot is not able to be removed fully. But in an angiogram picture, they may look very, very similar. We have also looked at a lot of different in vitro technique testing and modeling to understand how to most effectively use the devices we have. Just as an example, as really one slide that could probably take up 200, but we've done lots of work on this and understanding how the clot interacts with the catheter, for example, how different retrieval techniques might behave and be effective or not effective possibly, and looking at the flow visualization of and the flow dynamics in different conditions and using different devices. So this really gives us an appreciation for all the minute qualities. So in conclusion, I think that different clots really present different types of challenges, and the more really we can make our clots better, the better we can learn from it, and greater understanding can lead to better solutions. So this in vitro modeling can be a really valuable tool, but the output of what we get is really only as good as the input, and we feel a clot is a really key input in this situation. And there's so much more to learn on how clot can be impacted and the techniques can be impacted, and this in vitro modeling has the potential to analyze all these situations in parallel and build better models, and then find and understand optimal strategies that we might be able to adopt and when to use them, and therefore ultimately really better outcomes for the patients. Thank you for your time.
Like, how small can you now track the distal embolization? What is the smallest particle you're looking at?
Yeah, I mean, our lab doesn't have that microparticle capability, like, for example, Matt Kunis's lab has, and yeah, his lab is great, and he's got the capability of measuring up to the micron level. Definitely around the 10 to 20 micron level I've seen over there. So
based on what you describe, what is the current strategy that you think has the best chance for success on mechanical thrombectomy?
That's probably another 10-minute talk. But
yeah, just a simple question. Are you telling me that, because Matt will say to you in the end of his talk, he'll say it's balloon guide, intermediate catheter, and stent retriever. Do you agree with that?
I think that's because you don't often know what clot you're dealing with in advance. The best strategy seems to be to try and attempt for the worst situation, and we find that balloon guide, which causes the flow to stop, and an intermediate catheter, but an intermediate catheter used, for example, to pinch the clot rather than fully engulf the clot, because that interaction between the clot and the catheter we see quite a lot. And for example, if depending on the catheter being used, it could fragment the clot, or it could cause it to slip off because it's so fibrin rich. So in those cases, it's not just about the devices being used, but the technique and the nuances of it that make it beneficial.
Two questions. First of all, I don't know whether you have taken consideration the presence of von Willebrand factor in those clots. Well, published last year a paper showing that they play big part, a big role in the formation of the stroke clots, and they become resistant to TPA. The other thing is whether in these systems you can really do what happens in vivo, because I think that the nets and the neutrophils and other cells play very important role in the formation of the class. And we have paper ready to go to publication on this topic, that I don't think that those things could be simulated or done in vitro. And how could you take these things in consideration? Thank you.
Yeah, thank you. So there are, in addition to von Willebrand factor, there's lots of other proteins that are involved in clots that possibly we don't capture so well, because all the retrieved analysis of all the clots that are found in the literature that I've looked at, maybe more around 400 clots in total. So we tried to create our clot analogs based on the most abundant data, which is using H&E staining to find the red cell, fibrin, and white cell components. And those are really the data that we have in the literature that we were able to use when we started creating them. And probably this is a step ladder. And once we create a model, then we can add more functionality to those clots, as you suggest. But probably creating a model which incorporates nets and ischemic changes and those things is quite complex. And it's all about building a model and then stepping slowly up. Can't do everything at once, I think.
Yeah, but von Willebrand factor was described to be the major component of about 40% of the blood clots in stroke, in ischemic stroke. So I think the very important component that should be taken into consideration as soon as possible.
Certainly, I agree. Von Willebrand factor is an important component.
Thank you.
Thanks. I want to know, how is it possible to determine the nature of the clot before deciding the correct extraction procedure?
I think that really is the ultimate question. We've got several collaborations with universities in France, Germany, and in other areas that are looking at CT attenuation or MR sequences that might be able to distinguish clot characteristics or clot composition. Particularly maybe the amount of hemoglobin, or so there is research in there, and there was a recent meta-analysis by Walid Berjinki from Mayo Clinic that looked at how, among other things, how imaging relates to composition, and he found basically that there's the data is scattered all over. There's no one link, but the only thing he did find was that the hyperdense middle artery cerebral sign is strongly linked to higher red cell erythrocyte content, which is also linked to easier and faster retrieval or more successful retrieval. That's really the only link that's been made so far. I'd say that even when I was showing those pictures of AFib clots, for example, you could see a red cell rich clot and then a dark region on top, suggesting that the clot is not homogeneous and very heterogeneous. So which clot comes off, which piece of that clot comes off, and how do you detect it is still a question for understanding.
Please, I suggest you, when I remove the clot in the patient, I have the difference with the patient is treated before with fibrinolysis, intravenous fibrinolysis, and I don't know why the patient is not treated. It's not easy to remove, compact the clot, but when the patient is treated with by fibrinolysis, sometimes the clot is ruptured. Is it possible to test this in the laboratory?
We actually are beginning to look at interaction with TPA, and TPA relies on inherent plasminogen in the system to convert to plasmin and then cut up the clot. So doing it in vitro has some challenges because you need coagulation proteins inside there that are active. So there are some challenges associated with it, but we have tried to model those clots using the higher red cell rich content clots that have a less fibrin structure, more prone to fragmentation, and that's exactly what we do find that you saw in some of those videos where the red cell rich ones really break up, and we find that their weak fibrin structure can be difficult to do, and that's part of the reason why balloon guides seem to work very well in that situation. At least in vitro. All my answers are in vitro. Thank you.